Whenever a non-pathogenic antigen, commonly called as allergen, binds to the B cell, the type 1 hypersensitivity reactions or immediate hypersensitivity reaction starts. The common antigenic allergen includes animal dander, chemical additives, bee venom, breech pollen, raygrass pollen, codfish, and drugs like penicillin. It is called as immediate hypersensitivity reaction because it occurs within approximately 20 minutes of antigen exposure. During the type 1 hypersensitivity reaction, the allergen antibody complex on the surface of B cell is taken up, processed, and presented on a class II MHC molecule to helper T cell or Th cell. Here, the B cell represents the antigen to Th cell. The Th cell then binds to the MHC class II molecule on the surface of B cell through its receptor called TCR. The protein called CD4 associates with TCR and assists in the binding of TCR to the MHC class II molecule and antigen. The Th cell then produces a pleiotropic cytokine called IL-4. The glycoprotein IL-4 stimulates B cell to produce two types of cells. One is memory cell and another is plasma cell. The memory cells persist in the body of individual and can be converted anytime into the effector cells by subsequent exposure to antigen. The plasma cell then produces antigen-specific IgEs. The mast cell's surface has many high-affinity Fc epsilon Ri receptor for the IgEs. The antigen-specific IgEs then bind to the high-affinity Fc epsilon Ri receptors. The antigen-specific IgEs also bind to high-affinity Fc epsilon Ri receptors on the surface of basophil cell. Mast cell coated by antigen-specific IgEs are said to be sensitized. The same is true for basophil cells. When the same allergen is exposed second time to the membrane-bound IgEs on the sensitized mast cell and basophil cell, the IgEs are cross-linked. This IgE cross-linking causes degranulation of these cells. Due to this degranulation, the granules of the mast cell releases pharmacologically active mediators called as vasoactive amines. Histamine is one of the most important primary mediators of type 1 hypersensitivity. Other primary mediators exocytosed by mast cell and basophil cells includes proteoglycans like heparin, serotonin, carboxypeptidase A, cathepsin G, ECFA, NCFA, and natural serine proteases. The secondary mediators exocytosed by activated mast cells and basophil cells are leukotrienes like LTD4, LTC4, LTB4, prostaglandins, mainly PGD2, platelet activation factor or PAF. The vasoactive amines causes smooth muscle contraction in skin, constriction of intestinal and bronchial smooth muscle, dilation of small blood vessels, increased vascular permeability, increased production of mucus from mucous gland, platelet activation and aggregation. Vasoactive amines also stimulate sensory nerve endings. The vasoactive amines also increases the production of eosinophils. Widespread activation of mast cell leads to systemic effects of circulatory shock, hypotension, collapse, chest tightness, and in most severe cases, respiratory arrest and death. This is anaphylactic shock. Please like, subscribe and share.